welcome to my presentation of the structure of skeletal muscle, and my name is Jordan Lujan. So let's get started. So before we get into skeletal muscle, I just wanted to point out there are three main types of muscle: smooth muscle, cardiac muscle, and then skeletal. So smooth muscle and cardiac muscle are both involuntary. And smooth muscle you'll see in internal organs, such as the digestive system, and cardiac muscle. The name cardiac means it relates to the heart, so heart muscle. And skeletal is the voluntary muscle. It allows you to move. So here is another image that shows skeletal, cardiac, and smooth muscle. The top row uh, would be something that a histologist might see under a microscope potentially, and the bottom row shows those images just with the more cartoonish representation superimposed on top of it. Alright, let's move on. So looking at skeletal muscle, we have these your organ of muscle there attaches to a tendon, which then attaches to the bone. The bone's there just for some, structure, support, and everything, and the muscle is what moves all of that around. And so let's look at those different parts of the muscle. So let's start by looking at a muscle fiber generally. A muscle cell, also known as a muscle fiber, they are cylindrical for skeletal muscle, multinucleated, uh, meaning there's multiple nuclei per cell, per fiber. There are striations, um, and those are caused by the myofibrils. There are many mitochondria, and that is because ATP is needed for every movement. A lot of ATP is required. And then you have the sarcolemma, which is the membrane, and we'll get into some of the other organs as we move on. So I just wanted to point out for multinucleation, the reason these cells have multiple nuclei is because the myoblasts, the forming cells, will fuse together, and eventually in a mature cell you will have multiple nuclei. So here are some layers you have to a muscle. So of course you have your bone, your tendon, and then on the outside of the muscle you have a layer, the epimysium, and then going in from the epimysium you have other bundles, you have some blood vessels in between, and the other bundles that you have would be like the perimysium, and within the perimysium you have fascicles that hold um, a bunch of fibers, a bunch of cells in there, and between the fibers you have endomysium. So epi meaning upon something, peri meaning like around, and endo meaning within. So more or less that's what those prefixes mean. And so let's move on. So now here are some other structures within skeletal muscle. You have T-tubules. Those are represented by the yellow kind of tunnel looking things, and they're around myofibrils. They allow for action potentials to travel. Whenever a nerve cell sends a action potential to make the muscle move, the T-tubule is what allows it to start doing something. You have the sarcoplasmic reticulum, contains calcium ions, and you have the terminal cisternae, which allows for the movement of those, and The terminal cisternae is represented by the blue, the light blue that kind of wraps around those fibers. And then you have the sarcoplasmic reticulum, looks almost like a net. I believe that's what reticulum means. So now looking at these myofibrils, these proteins within muscle fibers that allow for movement to occur. Um, so you have thick and thin filament fibers, and these are different cytoskeleton elements. You have the thin filament, which is actin, and you can see the actin little bunches there, little circles, the proteins. And then you have the thick filament, the myosin, and the myosin is what's in the middle um, around the M line. And so a sarcomere is what we are looking at here. A sarcomere goes from one Z line or Z disc to the next. So that would include one Z disc, a M line, and then another Z disc or Z line, depending on your source, the preference of that. And a sarcomere is the smallest contractile unit of muscle. And there is another protein in here. Down in the bottom left of the picture, you can see where it's pointing at it, titin, or titin. I've heard it pronounced both ways. It's a elastic protein that helps for the stabilization of the sarcomere. So let's move on. So with our movement, you have a cross bridge formation. Um, this is where the myosin will bond or bind with the actin. So myosins have heads and tail regions, and they are intertwined. A myosin is intertwined with another myosin. So you have like a double head sort of and a wrapped tail. And on the head region, there is two major sites, an ATP binding site and an actin binding site. Um, the ATP binding site is used to make the myosin move and attach to the actin, and then another ATP will come along and will allow for detaching to occur. Also, when the ATP comes, it will go through the process of hydrolysis, which will break it down to ADP and a inorganic phosphate when the ATP attaches to the myosin head. Actin. In the actin subunits, you have tropomyosin, which will block the binding sites. But you have another protein called troponin, which has a calcium binding site. And when calcium comes and binds with troponin, it'll move the tropomyosin out of the way. That way, a myosin head can attach to the actin and a muscle contraction can occur. So here is the big picture, all that information kind of put together. The big picture, whenever you have a motor unit, you have a neuron and a muscle fiber come together. You have a neuromuscular junction. The neuron, the axon terminal from a neuron will come and meet with the muscle, and it will release acetylcholine, a neurotransmitter, through vesicles, and that will send a little bit of a action potential that will travel down the T-tubule, and the T-tubule will release calcium ions from voltage-gated channels. The calcium will travel down and eventually be released into a sarcomere region where the calcium will bind to the troponin on the actin, move the tropomyosin. Binding sites will then be exposed for myosin to attach, and it will pull towards the M line. This is called the sliding filament theory. So the sarcomere will shorten, not extend. In a relaxed state, it's already extended. So um, whenever the sliding filament theory is in action, the Z lines will move towards the M line. And so that is how a muscle moves. So that is the general structure of skeletal muscle and how it kind of works. And that is all. So here are my sources, and thank you for watching.